Hey everybody, welcome back to another video. Today we're going to talk about glucose homeostasis, and there's a couple of important organs involved here. So before we get into this, guys, if you liked the video, if you learned something from it, please go ahead and like, comment, subscribe. This video is going to be part of the nutrition and human metabolism playlist. Let's get started. Okay, so when we start talking about glucose homeostasis, there are a couple of important organs involved here, okay? So one of them is going to be this guy here, the liver. The next one's going to be the stomach. And of course, the small intestine is involved. But the main guy here is going to be the pancreas. There are other tissues like the skeletal muscle, the adipose tissue, and we'll talk about it, and a little bit the kidneys as well. But the pancreas is big here. So first things first, we got to talk about the pancreas. So if you guys remember from the digestive system video, which I'll link here if you haven't watched it, we talked about an overview of the pancreas, and we talked about the fact that the pancreas has the acini and the islets of Langerhans. But you know, the islets of Langerhans, they actually have more than just those two cells. So we talked about the fact that they have alpha cells, right? We talked about the fact that they have alpha cells, beta cells, but they actually have more than that, okay? They also have things like delta cells, they have pancreatic polypeptide cells, they also have epsilon cells, okay? Now, what do all of these guys release? Well, the beta cells, they release insulin. The alpha cells, they make a hormone called glucagon, okay? The delta cells, they make somatostatin. Somatostatin. Pancreatic polypeptide cells obviously create the pancreatic polypeptide. Very creative, I know. Polypeptide. And of course, the epsilon cells make ghrelin. Ghrelin. Now, you might be asking, hey, Keon, didn't you say back in that gastric video that somatostatin actually comes from the stomach, and ghrelin also comes from the stomach? Well, you'd be right. It actually does come from the stomach as well, but just so you know, somatostatin is an inhibitory hormone that is made in multiple places, okay? So it can be made at the level of the hypothalamus, sometimes called the growth hormone inhibiting hormone. It can be made here in the stomach. So if you guys remember, in the stomach, specifically at the gastric pits found in the oxyntic mucosa, you had these D cells, and these D cells will actually making somatostatin, okay? And ghrelin, at the level of the stomach here in the fundus, specifically in the, again, the oxyntic mucosa, okay? At the fundus, you have specific cells called P/D1 cells that make ghrelin, okay? Cool. I just wanted to let you guys know that other places make these kinds of hormones. Okay, but why should I care about all these different types of cells? Well, today we're only really going to talk about two of these. We're going to talk about alpha and beta. But do note that all these other different cells, they play an important role in the pancreas. For example, somatostatin secreted from the delta cells actually inhibits both alpha cells and beta cells, okay? So it inhibits both of them. So it tightly regulates glucagon and insulin secretion levels, okay? But we're not going to talk about that today, okay? So just a bit of note on what is the percentage of these cells within the actual pancreatic acini, okay? So the beta cells are about 70 to 80% of the islets of Langerhans. And I misspoke beforehand. I said the pancreatic acini. I meant the islets of Langerhans, okay? So the beta cells take up about 70 to 80% of the islets of Langerhans. Their alpha cells are about 20 to 25%. And you can actually say that the remaining 3 of these contribute to about 5 to 10% of the actual acini. Not the acini, sorry, the islets of Langerhans. Okay? So again, one more time, the islets of Langerhans have all of these endocrine cells, whereas the pancreatic acini contain those digestive enzymes secreted during digestion and absorption. The islets of Langerhans consist of beta cells, alpha cells, delta cells, pancreatic polypeptide cells, and epsilon cells. These 3 together make up about 5 to 10% of the islets. The alpha cells make up about 20 to 25%, and the beta cells make up about 70 to 80%. So when is insulin secreted? Okay, that's when we want to know. What is insulin and when is it secreted? So insulin is a peptide hormone, and it is actually released when blood glucose is high. So blood glucose is high. That's when insulin is released. Glucagon is actually released, and this is actually very easy to remember, glucagon. So it's released when glucose is gone. When glucose is gone / low in the blood. Okay? And we're going to look at an approximation of when it would be released. Okay? All right, now that's that. Now, from the small intestine, there is going to be 2 hormones that I want you to know. If you recall, the small intestine had these enteroendocrine cells, right? We're going to only talk about 2 of them. If you want to know the full list, again, the gastric video is linked here, so you can go and watch it. We had the L cells and the K cells. Now, the L cells, they were actually making something known as GLP-1, glucagon-like peptide 1, and these K cells were making GIP, okay, so glucose-dependent insulinotropic peptide, sometimes even called gastric inhibitory peptide. And again, in the GI video, I mentioned why it's no longer called the gastric inhibitory peptide. Okay, so that's going to be that. Now let's get into 2 different scenarios. We're going to talk about glucose homeostasis in a fasted state. Okay, so for example, an overnight fast right after you wake up in the morning. And then a fed state. So for example, right after you eat breakfast in the morning. Okay, we're going to look at how things are differing here. Okay, so already you're going to notice a whole bunch of different tissues here. You're going to notice the brain, the skeletal muscle, the adipocytes, the pancreas, and the liver. Okay, and this right here is just a vascular connection connecting them all together. All right, let's get into it. So, first things first, you are in a fasted state, okay? Meaning what? Meaning you are not eating right now, so you're not getting exogenous glucose into the bloodstream, okay? So what has to happen? Let's assume that your blood glucose drops, okay? And let's say that it drops to around 3.9 millimoles, okay? Because you guys know that the regular amount of glucose in the blood is about 5 millimoles per liter, but for now, let's assume that because you're in a fasted state, your glucose actually dropped to about 3.9, okay? And let's take it actually one step further. Let's say that we were actually at 4.5 millimoles, okay? Which actually, at 4.5, endogenous insulin secretion from the beta cells starts going down, okay? So at 4.5, we actually inhibit the beta cells from making glucose. So what's the stimulus here? Here's the GLUT2 transporter where glucose should be coming in, but we're in a fasted state. So glucose is actually not going to be coming in. What does that mean? That's going to actually stimulate the alpha cells to make a hormone called glucagon, okay? They actually make a hormone called glucagon. And it's actually going to be released at approximately 3.9 millimoles of blood glucose concentration. So this glucagon that's released, it'll go throughout this hepatic portal vein, okay? And once it gets to the actual liver, it's going to do a couple of things, okay? It's going to do a couple of things. So first things first, First things first. I got to talk about the different processes that are involved in the liver. You guys know the liver, it is the main organ responsible for maintaining blood glucose levels, okay? The liver is able to go through a process known as gluconeogenesis. Gluconeogenesis refers to the process of taking non-carbohydrate substrates and converting them into glucose. What kind of non-carbohydrate substance? Well, things like lactate, which is a byproduct of anaerobic metabolism, glycerol, which actually comes from the breakdown of fats. We'll actually see that and link it back here. And amino acids like alanine and glutamine, okay? There's other ones as well. Now, that's what gluconeogenesis is, okay? So taking these kind of substrates, I can actually produce glucose. I can produce glucose. You know what glucagon does? Glucagon says, hey, liver, currently the blood glucose levels are at 3.9. I want it to be at 5.5. So I'm going to stimulate the liver, okay, to go through gluconeogenesis and make glucose. And once this liver actually makes glucose, you know what it's going to do? It's going to push it out into the bloodstream, okay, to maintain blood glucose concentrations so tissues can get proper glucose amounts, okay? Proper glucose amounts. Does that make sense? Awesome, guys. So, what else does glucagon do in the liver? What else? Well, you know, in the liver, we also store glucose, right? So if you remember from the digestion and absorption of carbohydrates video, which I'll link here for you guys to watch, we said that the liver takes the glucose that it absorbed and turns it into something called glycogen. Okay? Glycogen is a storage form of glucose. Now, let's move this here. This glycogen can actually be broken down into glucose in the liver. You know what's the process of breaking down glycogen into glucose? That's actually called gluconeogenesis. So glycogen, okay, because I'm taking that glycogen. Lysis means to break. So glycogenolysis, okay? I'm breaking down glycogen. Cool. So what does this all have to do with anything? Well, our good friend glucagon is going to also stimulate this to happen. Glucagon is going to stimulate the conversion of glycogen in the liver to glucose. And we can also release that into the bloodstream as well, okay? To maintain blood glucose concentrations. Okay, that's cool. What else? What else can happen? This glycogen as well can be used, to form pyruvate and go to the TCA cycle and the electron transport chain to give energy to the liver itself. And obviously this process is called glycolysis. Okay? Awesome, guys. So, that's what glucagon is doing. But you know glucagon is doing another thing? You might be thinking, hey, Keon, where are we getting all of these amino acids you mentioned, like alanine and glutamine? Well, you can get them from the cell, right? You can get them from the liver cells. You guys know what liver cells are called, by the way? Liver cells are called hepatocytes, okay? So liver cells are called hepatocytes, okay? So these hepatocytes, they can get these amino acids by breaking down proteins inside of them, or they can actually uptake them from the bloodstream. So you know there's going to be amino acids in the bloodstream, and they have to come in through a specific transporter, okay? I'm not going to talk about that in this video. That'll be for protein digestion and absorption. But how do they get in? You know, under fasted conditions, glucagon is going to actually stimulate amino acid uptake into the liver, so we can use it in a process of gluconeogenesis. Do you see how beautiful this is? Glucagon is stimulating multiple different steps for us to actually be able to maintain blood glucose levels. Now, one little caveat I want to add here is that technically the liver is not the only organ involved in maintaining blood glucose homeostasis, okay? I'm sure you guys have heard of this bean-shaped organ we have. Let me draw it a little better. The kidney. The kidney is also involved because it's the other organ. It is the only other organ that can go through the process of gluconeogenesis. However, the liver cannot do the kidney cannot do glycogenolysis. It can only do gluconeogenesis. And what it does is, it's also going to produce glucose and put it into the bloodstream. Okay? And that's also going to be stimulated by glucagon. Now, again, usually we say that the kidneys have a minor contribution to this, so we won't talk about it too much. Okay, so that's how the hormone glucagon released from the alpha cells of the pancreas is working to keep blood glucose normal during fasted conditions, okay? What happens to glucagon once it actually leaves this liver and circulates to these other tissues, okay? What does it do there? Well, you guys know that all of these tissues are capable of taking up glucose. So for example, here's glucose. It goes through these GLUT1 transporters at a basal scenario. And that glucose, for example, can be used. It can be used for energy, okay, by this adipocyte. But they actually have a very low, both the adipocyte and the skeletal muscle, they have a very little need for glucose in fasted state because they actually use fat as a substrate for energy. They actually use fat as a substrate for energy. That's why. Okay. So what's happening here? What's happening here? Let's see. Let's see if I can point it out for you guys. You know, inside of the adipocytes, we actually store a lot of fat. And we store it mainly in the form of something known as triacylglycerol. Briefly, what does a triacylglycerol look like? So, glycerol is just this polar hydrocarbon chain molecule, okay? And attached to it is going to be fatty acids. So here's one fatty acid, here's another fatty acid, and here's another fatty acid. Because there's 3 fatty acids attached to it, we call it triacyl, right? You can think of this as acyl, and glycerol. Okay? Glycerol. I can't type. Okay. So this is a triacylglycerol. Now, within these adipocytes, there's going to be a specific enzyme. This enzyme is called hormone-sensitive lipase. The name should be pretty self-explanatory here. So this hormone-sensitive lipase, it's a lipase, meaning what? Meaning it's going to cut lipids. So it's going to take this triacylglycerol, and it's going to cut it, and it will produce 2 things, okay? It'll produce a glycerol molecule, and it'll produce a non-esterified fatty acid. What does this mean? What does an esterified mean? What does esterified mean? Very briefly, the bond between this glycerol molecule and the fatty acids is called an ester bond. Okay? And so when I cleave that bond and I cut it, I release something called non-esterified fatty acids. Okay? Because previously they were actually esterified, but now they're not esterified. So we have non-esterified fatty acids and glycerol released once this hormone-sensitive lipase cuts the triacylglycerols. But why is it doing that? Why is it doing that? Well, the hormone glucagon is stimulating this, okay? You know what this process is called? When I actually cut fat, right? When I start cleaving fat and using it, that's called a lipolysis. Remember what I told you about lysis? Lysis means breakdown. Lipo refers to this lipids. So lipolysis is lipid breakdown or fat breakdown. So you can see how during the fasted state, we're actually using up fat as a source of energy. So this fat can actually be used as a source of energy, right? It can go through a process known as beta oxidation and then go to the TCA cycle, which will then go to the electron transport chain, okay? But that's not the only thing that this non-esterified fatty acid can do. This non-esterified fatty acid, it'll go out into the blood, okay? Into the blood. And over there, it'll be bound to a specific carrier protein known as albumin. So here's albumin, and it's binding this non-esterified fatty acid. Okay, and this albumin is going to carry this fatty acid to organs that actually need energy. Okay, organs that need energy. So, for example, it will travel to the skeletal muscle, and it'll let go of this fatty acid, non-esterified fatty acid, and it can actually move into the skeletal muscle that way. Well, if it's doing that, it has to move through some sort of transporter. What is that transporter? That transporter is called CD36, okay? So the CD36 transports fatty acids into the skeletal muscle. And again, once this fatty acid comes into the muscle, it'll do the exact same thing. It'll go through the process of beta oxidation, and then after that, it'll produce a molecule called acetyl-CoA, which can then be put into the TCA cycle, which can then be used to go to the electron transport chain and make ATP. Okay. And you guys also know that from the bloodstream, we might be getting a little bit of glucose through these GLUT1 transporters, which can also be used for energy production. But I want you to know that the main way during the fasted state is actually that we're using fat. We're using fat. Now, technically, the muscle does have stored glycogen that it can break down into glucose. But again, at a resting state or in a basal state, for example, when you're sleeping and you wake up, we're really relying on non-esterified fatty acids, okay? But this can also happen to a small extent. Okay, so we talked about the fate of that non-esterified fatty acid. What about the fate of this glycerol? Well, you guys remember I told you glycerol can actually go to the liver. It can go to the liver, and it can be also used for a process known as gluconeogenesis. We already talked about this, guys. We already talked about this. Here's that glycerol. It can be used for gluconeogenesis to make new glucose, to then be put out into the bloodstream to maintain blood glucose levels. Why am I maintaining blood glucose levels? What am I doing? Why am I doing that? I'm going to write a quick note for you here, okay? That during the fasted state, because that's where we are, the brain, the gut, and the red blood cells have about 75 to 80% glucose utilization. So majority of the glucose that's actually being utilized, 75 to 80% of that glucose, is actually by the brain, the gut, and the red blood cells. Okay? This should make sense to you as to why the skeletal muscle and the adipocytes are actually using fatty acids for energy production, right? Because there are other things more important that need glucose. For example, the brain. The brain cannot use fat as a source of energy because fat cannot actually go through the blood-brain barrier. So it needs glucose. Now, technically, the brain can actually adjust to using a molecule known as ketones, but that's for another video. Okay, so that's how glucose is being used in the fasted state. And of course, again, in the brain, we've already talked about this. There's specific transporters. At the blood-brain barrier, glucose has to go through GLUT1 transporters. And if it wants to get into the neuron, it has to cross through these GLUT3. Once in there, it goes through the Krebs cycle, which will then go to the electron transport chain and make us ATP. Awesome. Okay. So last thing I want to mention here is that there can be neural regulation of the pancreatic alpha cells and beta cells. Okay, so for example, when the parasympathetic nervous system and the sympathetic nervous system act on this guy, this alpha cell, they're both actually going to stimulate the release of glucagon. Okay? Neither of them will actually inhibit it. They will both stimulate it. We'll talk about the beta cell regulation a little bit later in the next slide, actually. Okay? And obviously, once glucagon has done its job, okay, and restored blood glucose from, that 3.9 to 5, then glucagon release will then be inhibited again, okay? And there's this constant cycle of maintaining blood glucose at this 5 millimoles, okay? It's very much finely controlled, okay? And it's very much so done by the liver, okay? Because they are able to go through gluconeogenesis and glycogenolysis, okay? This glycogenolysis is breaking down the glycogen into glucose and releasing it out into the bloodstream. And gluconeogenesis is making glucose from non-carbohydrate substrates, okay? So that's how we actually have glucose homeostasis in the fasted state. What about in the fed state? So I just woke up, okay? I was in the fasted state, and then I had my food, my breakfast, and now my blood glucose has gone up, okay? And on top of blood glucose, other things have gone up as well, okay? So what's happening there? How do I regulate that? So like I said, when you eat, right? For example, when you eat breakfast, okay? Let's say you had some eggs with some bread. Obviously, the bread had some carbohydrates in it, so that's going to give you glucose, and the egg has proteins in it, so that's going to give you amino acids when you break them down. So the glucose will obviously circulate and come to this pancreas and stimulate the beta cells to make a special hormone, and we all know this special hormone here. This special hormone is called insulin, okay? So we make insulin. Awesome. And by the way, the presence of insulin actually inhibits glucagon secretion from the alpha cells, okay? So it actually inhibits glucagon secretion from the alpha cells. But glucose is not the only thing that can stimulate the secretion of insulin. Amino acids can do the same thing, right? So those amino acids from the egg you just ate, they can completely do that, okay? And what kind of amino acids? For example, leucine, arginine, lysine, right? They can all do that. They can all stimulate the beta cells to make insulin, okay? Another thing, while you eat, your food is going through the stomach, okay? And once it reaches this small intestine, you had those special cells there. Remember? The K cells and the L cells that we talked about? Those guys are going to be important as well. They were actually secreting hormones like GLP-1, hormones like GIP, okay? And if you guys think back to the video that we talked about these two in the digestive video, we mentioned that these guys are incretins. They have an incretin effect, okay? I like to remember incretins like this. Incra for increase, and then ins for insulin. They increase insulin, okay? So let's follow these guys. Let's follow these guys right here, and let's see where they're going to go. They're going to circulate all the way towards this pancreas, and look at that. They're going to also stimulate the beta cells to make insulin, okay? Awesome. There's other things as well. Remember the neural regulation? Well, when you're in a rest and digest scenario, and you just want to digest your food, you just want to eat your food, okay? You're digesting it, rest and digest. That's the parasympathetic nervous system. That's going to stimulate the activity of these beta cells. But, when you're exercising, your sympathetic nervous system is pretty high. That's going to actually inhibit the activity of this beta cell. And this is exact opposite to glucagon. Remember, we said glucagon is stimulated by both the parasympathetic and the sympathetic nervous system, okay? And this is to ensure that insulin doesn't lower our blood glucose by too much, okay? Great. So that's about the neuroregulation as well. Now, the whole point here is we released insulin. What is insulin going to do? The first organ to see insulin, just like glucagon, is the liver. The liver sees insulin first. So insulin goes into the liver, and it's going to do a bunch of things, and believe me, we're going to follow that. But I have to mention a concept here. Because the liver is the first organ to see insulin, it actually is able to modulate the amount of insulin that makes it out to all the other organs. So the liver actually reduces circulating insulin by 40 to 50%. Okay, so the liver is actually exposed to insulin and glucagon levels that are 5 to 10 times higher than what other organs see. So the liver actually reduces insulin by 40 to 50%, and that's because of something known as the first pass hepatic extraction. Okay? First pass hepatic extraction, which means that insulin has its first pass through the liver, right? Which is, by the way, hepatic, right? Hepatocytes, liver, right? They're going to extract a lot of that insulin. And that's just going to, remain very little insulin in the blood afterwards, okay? Which is good, actually. That's what we want. Okay, now let's follow what insulin is going to do in the liver. What is insulin going to do? I want you to think about insulin as an anabolic hormone. You know we have anabolic and catabolic processes in our body? So anabolic, and I'm sure a lot of you gym goers know this, anabolic is to actually build up something. Build up. Catabolic is to break it down. Break it down, okay? And you could kind of classify glucagon as a catabolic. In the last slide, if we go back, you can see that it's breaking down the glycogen into glucose, okay? And it's promoting the breakdown of fat into glycerol and non-esterified fatty acids, which was called lipolysis. Insulin is the exact opposite of that. It does the exact opposite of that. So what do you think it's going to do? It's going to promote glycogen storage, right? It's going to promote making glycogen. It's going to promote making fat, okay? And we'll see that right now. So you eat some glucose, right? Remember from those eggs or something, and they're here, and they're going to make their way from the portal vein all the way up to the liver, right? They're going to go through that GLUT2 transporter. Once they come in, once this glucose comes in, what's going to happen? So here's the glucose, okay? And it can go through many different routes. If you guys remember, I talked about this beforehand. But in this scenario, things are going to be a little bit different. So it can go through something known as de novo lipid synthesis, which just means that you're taking glucose and you're converting it into fat in the liver. You're making fatty acids, okay, off of that. And that could absolutely occur. That could for sure happen. But something that's also going to happen is that the glucose is going to get turned into glycogen, okay? Or it can be used for energy production, okay, like pyruvate to TCA cycle and then to the electron transport chain, okay? We talked about this beforehand. Insulin is doing a couple of things, okay? So not only can insulin come here, okay, and stimulate the conversion of glucose into glycogen, okay, but it does something else. Remember the liver? It could go through a special process. It was called gluconeogenesis, right? After it did that, it would make glucose, and it would put that glucose out into the bloodstream. Well, insulin is saying, hey, listen, we have too much glucose in the blood right now. We don't need that much. So I'm going to go ahead and I'm going to inhibit gluconeogenesis. Okay? So it does that. What else? What else can it do? Well, and I'm also going to just show this here. Glucose to glycogen. It also has a separate name, so We said that glycogen to glucose was called glycogenolysis. Glucose to glycogen is known as glycogenesis. Genesis meaning making of something new. So we're making glycogen. Okay? So glycogenesis. Boom. Awesome. So that's what insulin is doing in the liver. Okay? That's what it's doing in the liver. But you know, majority of the circulating glucose, right? The liver doesn't hog all of it. I've mentioned this before. The liver is not greedy. It doesn't want to keep all that glucose. So majority of that glucose is going to go and circulate elsewhere. Okay? Circulate elsewhere. In fact, as I've mentioned before, the liver, it doesn't like to hog the glucose that is coming in from the diet. So in order to maintain its own glycogen storage, what it's going to actually do is it's going to go through gluconeogenesis, it's going to make glucose, and the glucose that it made from gluconeogenesis is going to be converted into that glycogen. So we can actually put an arrow here for insulin stimulating glycogenesis as well. Okay, awesome. So what is insulin's action and relation with glucose in all these other tissues, right? So you know, in the adipose tissue and the skeletal muscle, we have specific transporters, right? You know, we said we had GLUT1, but this GLUT1 is really used in basal states, okay? And it barely gives us any glucose anyways, because these guys don't need that much glucose in a fasted state. They use fat as energy. But during the fed state, they love using glucose, and they have to absorb glucose. So how do they do that? They have a vesicle inside of their plasma membrane that contains these specific GLUT transporters. It's called GLUT4 storage vesicle. Okay, so the GLUT4 storage vesicle. And it has to somehow make its way into the membrane where all of those GLUT4 can then be placed onto the membrane. You know what actually stimulates that process? Insulin. Insulin comes and binds to its receptor on the skeletal muscle and says, hey, GLUT4 storage vesicle, it's time for you to come to the membrane because I want you to start absorbing glucose. Okay? All right, great. Once that actually happens, the glucose, and let's write it in a different color here. Oh my goodness. The glucose will get absorbed through the GLUT4 transporter and it'll come in. Okay? Once it comes in, some of it might be used for energy, but the majority of it will actually be turned into glycogen. It'll be stored for later. And who's stimulating this step? Insulin. Insulin is stimulating that step. Okay? So that's what's happening at the level of the skeletal muscle. What about the adipose tissue? Well, adipose tissue, they also have this GLUT4 storage vesicle. Remember this GLUT4 storage vesicle? Right? We talked about it here. It is also going to translocate towards the membrane. And who's stimulating this? Insulin. Insulin is doing that. Cool. And once it does that, then we can take up glucose and use it in this fat cell. But you know what fat cells really do? Fat cells don't really store a lot of glycogen. What they do instead is they take the glucose and they turn it into fat. Remember what I said the main storage of fat is in the body, guys? If you're saying triacylglycerol, you're absolutely correct. So we take this glucose and through a series of enzymatic steps, we turn it into glycerol 3-phosphate. And we have fatty acids that are actually just inside of this adipose tissue. And we're going to combine these two together and we're going to make triacylglycerol. Okay? Who's stimulating this step? Insulin. Insulin is saying, hey, go ahead and make lipids for me. Well, you guys remember when we were breaking down lipids? That had a name. That was called lipolysis. What about when we're making lipids? That's called lipogenesis. So insulin has a lipogenic effect. Okay? Remember, it's an anabolic hormone. It wants to build things up. So it's building up fat. It's building up glycogen. Okay? And, for those of you who are inside of the gym space, you might also know that insulin promotes amino acid uptake into the muscle cell, which can be used for protein synthesis and making your muscles bigger. Okay, but I'm not going to talk about that because that's not related to glucose homeostasis. Now, you might be thinking, hey, Keon, you mentioned a specific enzyme in the last slide that was breaking down this triacylglycerol. That was called hormone-sensitive lipase, right? You said that it's going to act on this guy, and it's going to break it down into non-esterified fatty acids and glycerol. Well, how do we make sure that this doesn't happen now? Okay? Well, I'll tell you how we make sure that doesn't happen. You actually just completely inhibit it. What's inhibiting it? Insulin. Insulin comes here and inhibits hormone-sensitive lipase, so we're not breaking down the triacylglycerol, and we're continuing to make fat. Okay? So that's what insulin's doing. Oh man, that's awesome. Now, of course, glucose is also going into other tissues like the brain. Again, I've already mentioned this, so I'm not spending time on it. TCA cycle and then ETC. Okay, cool. The main parts here were just these skeletal muscle and the adipocyte. Okay. One thing you got to understand is that the skeletal muscle and the liver are the main sites of glycogen storage in the body. Okay, and actually, glycogen, when it's being stored, it's actually being stored with water. Okay, it's actually a 1-to-3 ratio or a 1-to-4. Okay, it just depends. But for every glucose molecule that I'm storing, I'm bringing in 3 waters with it. Okay, so it takes a lot of water. All right. Last thing I want to talk about here is that in the last slide we mentioned that the brain, the gut, and the red blood cells do about 75 to 80% of glucose utilization. And very quickly, you might be thinking, why red blood cells? Let me show you a structure of red blood cell, okay? Here's a red blood cell. Your red blood cell actually doesn't have a nucleus, meaning it can't go through protein synthesis. Not only that, it doesn't have a mitochondria. It doesn't have it. So it can't go through electron transport chain and the Krebs cycle. So what does it use for energy? Just glycolysis, right? And it can't go through using fat as well because this beta oxidation process occurs in the mitochondria. So it requires a high amount of glucose. It requires a high amount of glucose to go through glycolysis, specifically anaerobic glycolysis. Okay? So they actually make a lot of lactate. All right? As opposed to the brain. The brain actually doesn't make a lot of lactate at all. Anyways, that's why the red blood cells require such a high amount of glucose utilization. And the brain, like I said, they cannot use fat. They can't. So they have to use glucose, okay? And if you're very prolonged starving, okay, not just an overnight fast, and you're prolonged starving, you're not giving your body any glucose, your body starts to actually make ketone bodies, and the brain can actually utilize those ketone bodies. For example, some ketones could be beta hydroxybutyrate, okay, which is the most potent one. But there's also like acetone, there's acetoacetate, okay. These are just some of the ketones that the brain can use, but it's mainly, again, that beta hydroxybutyrate. Anyways, so what are the percentages here? Well, in a fed state scenario, the skeletal muscle contributes to about 85% of glucose utilization, and because it actually has to use this GLUT4 transporter, we actually say that it is 85% of insulin dependent glucose uptake. Glucose clearance, uptake, utilization, whatever you want to call it. Okay? It's the same thing. And the adipocyte contributes to about 5% of the insulin dependent glucose uptake. Okay? So compare that. At rest, 75 to 80% is being done by the brain, the gut, and the red blood cells, whereas during the fed state, about 90% is just with the skeletal muscle and the adipocytes. Okay? So that's big right there. It's very big. And one last note about this GLP and GIP-1, GIP and GLP-1. These guys are, like I said, they're incretins, and they achieve their effects by eating. Okay, so when you eat, these guys sense the food. Okay, they're stimulated by certain nutrients like glucose, like amino acids, and then they're going to be released by the K cells and the L cells from the enteroendocrine cells. But what if you're not eating? So, there are scenarios where some people, for example, are in the hospital and they can't eat, and they go through something known as parenteral nutrition, okay, which is just intravenous feeding. Think about this. If I inject into your bloodstream glucose, okay, am I going to get GLP and GIP release? No, right? Because it's going through the bloodstream already. It's going directly to the bloodstream. Instead of going through the esophagus, the stomach, and the small intestine where the GLP-1 and GIP are released, I'm immediately putting it into the blood. So I'm not going to get any of these. And if I'm not going to get any of these, I'm actually not going to get as high as insulin release as I would if I were to eat normally throughout my mouth. Okay? So just remember that when it comes to these incretins, okay? They modulate insulin secretion with in a concentration-dependent manner, okay? All right, guys. That's going to be everything for the glucose homeostasis video. If you like this video, please, again, like, subscribe. And if you have any questions, feel free to comment down below and I'll answer. Okay? That's going to be all of it. See you in the next one.